Pleasure, I guess, today to introduce Ken Storey. You'll see why I'm just guessing in a minute. He's a rather entertaining presenter, I think you'll agree. He's coming to us from Carleton University in Canada, where he is the professor of biochemistry and a Canada Research Chair in molecular physiology. Among other distinctions and awards, he is the fellow of cryobiology, which I didn't even know was a thing. So that was
you don't mention the Royal Society, you mention cryobiology. A bitter, bitter man.
Ken got his PhD under Peter Hochachka, who's widely considered one of the fathers of comparative physiology, and he was a professor at Duke University before moving to Carleton. I met him many times, but the first time when I was a postdoc, he visited Miami, but he doesn't remember that. And then I met him again years later. I gave a talk at a meeting in Denver that had something to do with metabolic suppression, which you'll hear a lot about shortly. And that sprung a collaboration where I took this man out to sea for a month, which was disastrous. The picture that's on the flyer going around is probably the best Ken looked on that cruise. He was miserably seasick, even though we were in the Gulf of California, which is dead flat calm the entire time. Couldn't even tell the boat was moving. That he was miserable. But we got a couple great papers out of it, and without further ado, I will let him explain the living dead.
And you still have to write up one of those papers on crabs, right?
Yes.
You lazy, lazy scientist. So, it's a thing. There will be marine animals in the end, but I'll cover the principles. I cover the principles of animals that are useful. Animals that are useful. Now, useful to me means that funding agencies love them. So medical funding agencies love these animals, and I had NIH grants for 15 years and NSF grants. So when you look at the animals I work on, figure out why I have DOD grants and heart and stroke grants and all sorts of other things, even though I'm not a practical human. So we're going to study animals that are alive. Sometimes they're warm, sometimes they're not, but they're alive. This is their basal metabolic rate. Then they become dead. Absolutely dead. They pull a plug on these. These are clinically dead. In a variety of ways, you'll see. You can get dead in a lot of ways. And then they come back to life. It's this part that's hard. This part is good. You freeze a house cat, pretty much this curve is exactly the same. We do use house cats as controls because I hate cats. So, all these animals are going to do this, from the most complicated and wonderful of animals, all the way down to the lowliest of sea creatures. Oh, have I lost the audience yet? So, I cannot use this thing because you've hidden it. You are so smart. He's so smart. I know. Still. Oh, good. This is a mammal, a warm, furry mammal, a funding trap for all sorts of different grants. This is a warm mammal that runs around. This is a mammal that's cold to the touch. This is a mammal whose body temperature is 5 degrees, whose brain has more or less completely shut off. This brain is filled with tau plaques, just like your grandma's brain is. But the difference is when this animal warms and wakes up, those tau plaques are going to disappear like magic. This is an animal that shuts down his heart and his liver. And many young people, bless their hearts, do really dangerous things. You idiots will do a sport where there's an ambulance at the bottom of the hill. What? And many of us ruined our organs in the 80s. So I'm just saying we need transplants. You people are very useful to me. Just keep healthy. The problem is you guys tend to die far away from where I need a heart. So my job, and huge amounts of taxpayers' dollars go to me taking a mammalian heart, making it live for a little bit longer so they can get it to me. So that's why this is important. and why, if we work out the principles in one animal, and if we're lucky enough to do them in all animals, it's going to be good. These are mammals. They haven't been ground squirrels in evolutionary time for a terrible amount of time. This is a bat, ground squirrels, bears, all mammals are probably capable of hibernation. We now work on a tenrec, which is supposed to be the basal mammal. The basal mammal is still around. It's like an armored mouse. It lives in Madagascar. It will do this live dead. So all the mammals we can find do this except you. Our theory is that you have all the pathways, but you can't organize them correctly. You are essentially the Brad Seibels of metabolic regulation. Your house is a mess, there's stuff. So, you know how sometimes you have stuff in drawers. In your house you have stuff in drawers, right? And you sometimes open the drawers so you can see what's in them. So what Brad does is he takes out a full drawer and he puts it on top of the table so he can see what's in it. His tabletops are covered with full drawers. It's just so wrong. He moved in June. You do the math. This is a primate, not one of the world's top primates, but it's a lemur. This animal and other types of lemurs are capable of undergoing torpor. This is a daily torpor genius, and they will stay in torpor for an entire week. If we could even keep your hearts better and in hibernated state for a week, it would change medical science entirely. I'm a member of what's called an Organ Preservation Alliance, and there's a large number of grants we run in the United States from coast to coast trying to look at organ preservation. But metabolic depression, the ability to shut yourself off and then come back to life, happened really, really early. This is an estivating snail. This one comes from Southern California. 10 years. You take away their water, they cover up, they breathe once a month. That's it. If you add water and stir, they come back to life. And what I want to assure you is every single molecular thing I'm going to force you to learn, and you're going to take notes about because there's a quiz, happens the same in every animal. Every animal is going to be the same. You learn this once you know it. And if you don't like invertebrates, which seems strange on this crowd, here's a vertebrate that does the same thing. A control spadefoot toad in Arizona is one under the ground. 11 and a half months a year, they're under the ground. They're dried, crispy critters. You pick them up, they're dry and crispy. You add water and stir, and they toad up. They mate in the ponds. It only rains once a year in Arizona. These things can stay under for 5 full years. They mate, they lay eggs, they smoke cigarettes, and they're down for the rest of the time. Their natural state is off. Their natural state is dried down. They have as much urea as a shark. Surviving without oxygen is something you'll recognize. The highest sort of vertebrate we study that can survive complete anoxia are turtles. They don't have brain problems, they don't have heart problems, but for 9 months they have absolutely no oxygen. And this is where the marine invertebrates come in. There's a lot of marine invertebrates that are very, well, we'd like oxygen, but if we don't have it, we're perfectly okay. The difference is with oxygen, without oxygen, with oxygen again. So they do the same tricks, and we'll show you the moleculars of the tricks. This is a frozen animal. This is a hoppy, squishy frog. It freezes solidly. Every water molecule in its body freezes down. Its blood is frozen. You have to collect it with a chisel. Its organs are all stopped because they're surrounded by ice. This is a cataract. This is the precipitation of proteins in their eyes because ice sucks all the water out. Their cataracts get better. They build up 400 millimolar glucose, which is enough to stun the best of the diabetics. You would certainly die depending on where 400 millimolar hits you. And therefore, these are a model of how you can survive as a vertebrate, not just frozen, but with hugely high glucose. They exist for months with very high glucose. Now, you would think, here's what you would think, isn't freezing enough? If you're going to freeze solidly, why the hell are you going to bother to do this? You're going to be frozen. Why don't you just wait it out? And the answer is frogs depress their metabolic rate first, then they freeze in order to stay dead. Think about how we transplant organs. We find a young person crumpled somewhere, probably on a motorcycle. Come on, things happen. Then we take out their organs and we cool them down. We immediately cool them down, just like a ground squirrel. But here's what a ground squirrel does instead. It turns off all of its organs, then it cools down. It cools down because it shut its metabolism down. So I want to tell you that thousands of things go on in every organ first. Then if you're a ground squirrel, you're cold because you're no longer making energy. So we're doing organ transplants wrong, and a group of us are trying to change that. Well, it's fairly hard. Mammalian hibernation. You look at their oxygen consumption, their CO2 release, you look at their ATP turnover, you look at how much amino acids they pick up, you look at what messages they're making and what proteins change, boom, they're out for the count. Their low body temperature as a result of being turned off, and the only reason why they have to hibernate is you never see herds of ground squirrels running through your town to go to Florida. If you're a bird, you fly. If you're a Canadian, you take a plane. 10% of the people you know are Canadian. We just blend in until the revolution comes. Oh, no, wait. No, I've said too much, eh? Ground squirrels go down for 2 weeks at a time. They go down and their body temperature drops to 5 or 6 degrees, and they turn off what looks like their organ metabolism. You can see these all in papers. What happens then is that every once in a while they have to break up. They have to warm up. The way they warm up is shivering thermogenesis, so we study their muscles. Their muscles are very special in that they can start shivering at low temperatures, but then also stop. You do not have to shiver if you're a ground squirrel. You're perfectly okay. Then they warm up with brown fat. So brown fat is an actual thing. They have brown fat that they warm up. So half of them shivers and half of them warm up with brown fat. And when they get here, what do? Do they run around? Do they go out skiing again? Do they listen to their iPods? No, they sleep. They sleep. When you have a cold mammalian brain, you have a brain that's jangled and horrible. It's like you spent those 2 weeks thinking you were studying biochemistry, but then you failed a test. You stayed up too long. These guys REM sleep up here, and their brain gets better, demonstrably better. It's not just tau plaques, it's also prion metabolism and a whole bunch of other things. So we take animals during their natural hibernation and ask, how are you doing? How's this working out for you? What are you turning on and off? How are you dealing with low? And then how do you warm back up? These are all important and all happen in all animals, no matter what the stress. And here's the first thing. 2 lines here, metabolic rate and body temperature. You would think that if you put a cold organism into the cold, you'd have it made. This is what we try to do with human organs. But any of you who have babies, they're too small to phone child services, put them in a fridge, close the door. There's some crying, some complaining, but then when you open them, they're still warm and grumpy. They are because you haven't turned off their metabolism. Mammals do not get cold and then slow down their metabolism. Metabolic rate falls like a shot, and then their body temperature falls. If you're little, your body temperature falls quickly. You cool like a small brick, like a large brick, like a huge frickin' brick. And as it turns out, if you're a bear, you never cool down. Their body temperature falls from 37 down to 30, but they're so big and they're so fat and they're so furry and they have so much fat that they never cool down beyond that. As it turns out, if I cool you down past 29 degrees, your heart stops automatically. These animals' hearts continue to go. And so heart and stroke are interested in these sort of things. Why don't they get strokes? Why don't they get heart attacks? So what happens in every case is you have to prove there's a metabolic rate reduction. That's why we have physiologists. They go out in the field when it's snowing, they put backpacks on ground squirrels, we make them do whole bunches of work for very low grants, and then we say, we'll tell you the metabolic basis, and they back up trucks. It's just a thing. What your body will do when I get hold of enough human hearts, And what every animal other than you possibly can do, first you turn on a couple things and they turn off everything else. So this is a series of enzymes. You have 750 protein kinases. Of those, about 10 turn on, and they reach out to the cell and turn everything else off. So this is like your dad getting up out of his chair and turning off all the damn lights. He turns off all the lights. Then what happens is genes are convinced to turn off. Not some of the genes, we're finished with that, but all of the genes that don't have a get-out-of-jail-free card. So most genes turn off, the only ones that stay on have a get-out-of-jail-free card. It's called an IRES sequence. Now that we've genome sequenced all of our animals, you can just run down and find out what they are. Once you are turned off, once you're dead to the world, once you seem to be dead, new genes turn on. Tick, 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 tick. They turn on in a frog that's frozen. They turn on in a ground squirrel. They're the same genes. A very few genes turn on, and that's to protect you. It's mainly to protect you from oxygen damage. It's mainly to protect you from these metabolic rate problems. You get glucose-regulated proteins and you get mitochondrial proteins. If you want to actually protect a heart for transplant, you have to do all of these things. It's not good enough just to turn it off. You've got to stimulate these genes at the bottom, which we regularly do in cells and tissues and organs. Why do you have to turn yourself off? Why do you bother doing this? Because hidden within your cells is machinery that is expensive. So, transcription. You are turning on and off 25,000 genes right now, and you're making messenger RNA. Every time you make messenger RNA, it costs you a fortune. If you do, then you have to make new ATP, then you have to burn down carbohydrates and fats, and you're hot. You turn off transcription. You stop your genes from turning on, all of them off. You save a ton of money, and you don't make any more heat. Then you stop making new proteins. You have to make new proteins or die. If I poison this in active cells in an active dish, those cells die. There's a number of different ways of doing it. So you have to turn off those expensive things, but guess what? Your cells are also doing really cheap things, and this is what your cell does do to turn yourself off. You turn this off with these, you turn this off with these, and these are expensive. There are some enzymes and some proteins that are only turned on functionally when you turn yourself off. So there's some things that are set to only turn on when you're off. These protein kinases are an example. Then you make those active, and then they reach out and turn everything else off. You have to stop breaking down proteins, and so what they do in the 3,000 pathways of a cell is they turn off most of them. This is what they're dancing with. You dance with them that brung you. This is what they're doing to begin with. And they're all doing it. If there's any one message in the many papers you will have to read, you'll have to PubMed me or you'll have to Google Scholar me. It's grotesque. If you're a hibernating ground squirrel, you get cold and you turn off genes and proteins and pathways. If you're a wet animal and you dry down, same pathways, same kinases. If you used to use oxygen and now you just don't care, same pathways. If you're freezing tolerant or cold tolerant, on your way down, same pathway, same enzyme, same everything. Everything is the same. It saves you on having to make new slides. It's excellent. Everybody does the same thing. So look, it's the same slide with an orange thing at the bottom. Now remember, all of these slides are on my webpage, so you can Google my webpage and find all these things. You don't have to worry about them. This is your cell. It looks as if it's been drawn by a grade 7 student, and it was. You are pumping ions. When you depress your metabolism, you have to stop that, and this is a very controversial area. Everybody is working on pumps and channels and things. You have to have your mitochondria on. You're sucking in small molecular weight molecules, you're running your TCA cycle, and unfortunately it's like running your mouth. If you make ATP, then you haven't turned off your metabolism. You're breaking down and synthesizing carbohydrates, amino acids, and proteins. You've got to stop that. You're turning on and off genes, you're making new proteins, and you've got to stop that. The way you stop it is right here. Cells listen, and their ears are two different kinds of kinases. And you don't have to worry about that, but I'll introduce them both because they operate on different principles. They're both protein kinases. They reach out and turn everything off. These little dotted lines are turning everything off. It's so cool. And it's a biochemical unity. So what we do, there's young people here, and your supervisors have sort of lied to you. Their theory is that you should take a course in the scientific method. And what you should do is you should have a hypothesis. Yeah. And then you should test that hypothesis. And then if the hypothesis isn't right, you should do other experiments. While you're doing that, I'm taking your grant money. You may as well be bringing lunch money and handing it to me, because do you know what actual scientists actually do? We measure everything. We've invented a word for measuring everything. Biochemists don't have to talk to each other anymore. We go, omics? And he says, dude, omics. That's it, we're done. We have proteomics. We measure all the proteins. We have phosphoproteomics. You measure all the phosphoproteins. We have microRNAomics. We measure all the microRNAs. Come on, there's only a couple of thousand. We have message RNAomics, or message RNA-seq. We have metabolomics. Honestly, we just measure all the metabolites. So it's me, a cup of coffee, a big-ass machine, and your parents' tax money. It's simple. We measure everything. So what we did was we thought, why don't we figure out which message RNAs would change? No! You measure them all, then the company sends you back and they tell you, and it turns out to be 99% of them are off. Well, what about proteins? They can't possibly turn off all their proteins. Why? You would die. They don't die, they turn them all off. They stop ion pumping, they modify the enzymes that used to pump ions and shut them off. They stop using carbohydrates. They switch over to fat fuels, but their metabolism, their overall metabolic rate is 1% of normal. You need oxygen. You need oxygen like you eat potato chips. You just can't do without it. You terribly cowardly things. If I was to do the right thing with Brad, he would be brain dead in 2 minutes. Okay, 1 minute for him because he's off the class. 5 minutes later, your heart would stop. These animals go, no oxygen, no problem, because they've shut themselves off first. If we could drop his metabolic rate further than it was at 2 o'clock in the morning when he crawled into, oh no, okay.